Thank God.
Yes, please.
Yep. Okay. Very good. So if we can move then to the third grand challenge topic, which is ecology and environment, and this is from Jiangxi Wang, Kevin Tyler, and Wade Huang. So could one of you please take it away?
Yes. So before we start, we want to know if Jiangxi is still here. Is here? Jiangxi? No, I don't think so. So can you see my share screen well?
Yeah,
There's the two of you. Okay.
Okay, so can you see my share screen? Hello?
Yeah, we can see your screen, please.
Okay, sounds good. Yeah, so we are going to talk about ecology and environment. My name is Wade Huang, and joining me today will be Kevin Taylor. So we all know that globally distributed Euglena are abundant plankton. They are members of which are able to occupy freshwater and marine environments as well as brackish water, and they are capable to do mixotrophic via photosynthesis and ingestion of particles. That evolutionary divergence and fresh as eukaryotes have some useful and high value chemicals as well as toxicants that can be damaging to the environment. The challenge in this area for ecology and environment are associated with understanding the exploration of different diverse niches, and then also peculiar biology and natural roles of that Euglena occupied environment. So to understand how research involving Euglena can lead to beneficial ecological impacts, this is how we pursue it, what we are pursuing. And Taylor and I are actually coming from two different backgrounds. So we are going to introduce our background to you from Taylor first.
Okay, hello.
Kevin, are you there?
Can you see me okay? Yeah, so just some people will know me, but I'm at Norwich Medical School and I'm a protozoologist for the purposes of this meeting. Somebody who's interested in structure-function relationships and in bioactivity and in the proteins that are under direct selection pressure that are associated with some of the activities which are important for Euglena function.
Okay, so my name is Wade Huang again. I used to be a PhD student and then did a postdoc with Paul Zimber. As you have heard earlier in his presentation, we mainly studied Euglena bison as well as some physiology changes in Euglena according to nutrient depletion, and we also outreach to health institution to do some cancer research on the using Euglena on other organisms. Currently, I'm working in Virginia Institute of Marine Science. My portion of my postdoc is using imaging for cytobio, which can do a rapid monitoring program to identify algae in the water, as well as Euglena. And my postdoc goal is to understand the interaction of all these algae in a year-long period. So we put together 4 questions for this section. The first question is to use Euglena as agents of remediation and to counter determinate environmental challenges. So basically, we want to know how we the detoxification or demineralization, as well as desalination and oxygenation of Euglena is actually a little bit of challenging for people to know right now. And as you heard yesterday, that's what studies involved in establish using the Euglena to absorb heavy metals such as cadmium. And also they were using them in the wastewater treatment as well. So further, we want to discuss how the development and reclamation and agriculture intensification programs. I think this was originally Wang's idea for this particular session. Excuse me, for this particular topic. The part that I can express on is on the spill and disasters about how to clean up the Euglena. For eukaryotes, we can use, for prokaryotes such as cyanobacteria, we can use hydrogen peroxide or copper sulfates to inhibit their growth and also inhibits the photosynthesis. But for a mesotrophy like in Euglena, it's pretty difficult as well. Particularly they are eukaryotes, so their cell wall is super hard to break down by hydrogen peroxide. So in order to use some enzymes or biological remediation technology, we also need to think about how to control the environment in an intact scenario. The next one is the interaction of the euglenoid and other algae. As I have described, I'm currently working with Imaging for cytobot that we can have a constant algal identification within every hour. And with these techniques that we don't have to spend a long time in front of the microscope. And then this way we will be able to identify the differences of the different population of algae in the water column with less effort. And the last part is how the climate change will affect the Euglena bloom in lake, even to the brackish environment. The second challenge questions that we have is to use the Euglena as a microbial factories for production of useful biomolecules and novel biopharmaceuticals. We know that Euglena are abundant in chemicals, such as we can use the Euglena to produce biofuels, wax esters, outgrowing paramelon, tocopherols, and unsaturated fatty acids such as DHA or EPA. We also know that in post presentation that Euglena produce a diverse scale of bioactive metabolites. Produce bioactive metabolites that can be used for cancer treatment, and this can open the routes to new natural compounds to exploit as well. The last one in this challenge question will be transgenic technologies provides potential of culturing bioactive compounds for other sources, such as drugs, like I mentioned earlier, for the cancer treatment. The third challenge question will be to use Euglena either directly or indirectly in the production of foodstuff. As we heard earlier with Noblegen's seminar, they were using the Euglena to do flowers. But however, Euglena also can be a carbon fixation sources, and so carbon fixation factors as primary producer in food webs. Also can use in aquaculture, food chains, providing biomasses or oxygenating water, directly as foodstuff for large-scale cultures, food and vitamin supplements, use of Euglena in aquaponics. Kevin?
So did you want me to take over here? So yeah, so we've been hearing from various sources, and Noble Jen made quite a case for the fact that Euglena is a force for good in the world, and so as we face all these environmental problems, that perhaps some of the answers can be supplied by Euglena, and the reason is that Euglena, in and of itself, unmodified, is basically a force for good. It already is a major carbon fixer, and it oxygenates when it's present in sunlight. And these are some of the things that we need to increase dramatically if we're going to combat climate change. So the idea that we could just take various euglenas and use them in different environmental niches is a strong motivator, and some of its characteristics as being able to sense its environment and to translate what it sensed into positional information for itself, and then to shift its metabolism as required, are useful characteristics to which potentially we can translate and exploit. So in this challenge question, we looked at whether we could use motile microbots, microscale rather than nanoscale, and exploit some of the things that some euglenas can do. Some abilities to sense magnetic fields, to know up from down, to sense light, or to sense chemicals in the environment, and to respond to these in an appropriate way. So Challenge 4 just lists some of the things that some of the euglenas are able to do. Almost all of them are useful to euglena, as you might expect. It's obviously evolved things to help it survive. A few things like toxin production, as we've heard earlier, can be negative features. And so taking on these characteristics, the sort of grand challenge. Do you want to move forward to the next slide, Wade? You've got it. Bit by bit. The question is, can we, using euglena as a sort of a model, slim it down from this massive genome and produce something which we could use as a microbot? And if you click the YouTube video there, you see is for the last 10 years, we've been able to isolate intact flagellum and make them swim. And the nice thing about flagella is that they're a constrained group of, let's go back to presentation. They're a constrained group of proteins with a very clear structure, which is pretty conserved, but their ability to towards or away from stimuli comes from subtle changes within the sort of core group of proteins. So we've been hearing about just how big the euglena genomes are and just how many proteins are involved at various stages. But actually, when you're thinking about the flagellum, you're only thinking of about 700 proteins or so. And in some flagellates, kinetoplastid relatives, which perhaps are less flexible in their ability to respond to the environment, but nevertheless, the structure-function relationships of the flagella are beginning to be quite well defined there. And so by comparative genomics, by comparative proteomics, and using some of the techniques that are arising in things like synthetic biology, the opportunity exists right now, I think, to undertake some sort of a bioengineering grand challenge to produce living microbots based on euglena that are able to do some of these things. And so I think just below is a picture of a flagellum with the paraflagellar rod, and you can see the paraflagellar rod and the axoneme quite well defined on the left-hand side, and on the right-hand side you get into this amazing spring-like structure of the paraflagellar rod, and it's from Euglena. See just how some of the tactic sensing and translation into directional taxis occur, and I think some more research in that area is very justified, but also we're probably in a position now that we can begin to think about making these microbots and carrying payloads which are environmentally friendly alongside, and so that's where I'll leave that with people for discussion.
Yeah, so I think we have one question. Was that an isolated flagellum responding to a stimulus?
Oh, so that was a clammy, sorry, this is all data from about 10 years ago. And that was from a Japanese group who basically took flagella from Chlamydomonas and grafted them onto beads and were able to show motility with them now. So that's a very, very primitive microbot. But I think that with some of the techniques available now through synthetic biology, we should be able to come up with much more interesting facsimiles of that.
Any questions? And that's the second one from Vladimir. Can we also use the ability of Euglena movements for microbots? Something like a micro pumping device, peristaltic micro machines.
So probably the person to answer that is somebody like Simone from yesterday. But there was some very impressive stuff there. I suspect that will make everything much more complicated. So depending on how simple you want to keep it and to keep the payloads. So there's obviously one can take the view now with the ability to CRISPR things, that we could be slimming these existing genomes and starting with euglenoids to produce essentially genetically transformed euglenoids to do what we want to do. But conversely, one could actually build from the ground up as that is a sort of main synthetic biology pathway where we actually create a bespoke euglenoid that does more or less what we want it to.
Yeah, actually, I think this sounds interesting. So I don't know if the Simonia is on the call to contribute to this. Is something?
Yes, I am.
Yeah. So do you have any comments with regards to this?
No, I was really very intrigued by the comments. I think a lot we need to know more. Just my instant reactions to Kevin Tyler's comments. So indeed, we know more about the flagellum than the mechanism for. Yesterday we were talking about the microtubules, the motors in the metaboly. We know more about the flagellum, even though we don't know everything. So there have been recent studies with cryo-electron microscopy, so that enable us to say a bit more. But I think the molecular mechanism is still unclear. It's very exciting prospect, but we need to understand more. I just wanted to resonate with how interesting these ideas are.
So we have a third question. Is there a company already using Euglena to remove any pollutants? As far as my understanding, I don't know, but is anyone in the audience that's coming from industrial side can give an answer on this?
This is the question of bioremediation and whether there is an existing company that's doing that. And obviously, NobleGen Scott's just saying, Noble-Gen started from that. But I don't know, Scott, if you want to say anything about.
Yeah, this is an area that we think there's opportunities in using kind of unique biological solutions to cleaning up the environment. So definitely, it's an area that we're interested in and seeking to find collaborations and also ideas. So very intrigued about the microbots and the peristaltic pumps. These are all cool ideas that I'm sure the Adam would find very interesting. So mostly just mining technologies at this point, but yeah, we're open to a lot of different cleaning up projects.
Yeah, so I don't know if Kengo is on the call as well. So I don't know if this is something.
Sorry, Singa?
Kengo. So I don't know if this is something Kengo and Kemi. I'm not sure. Yeah.
I think this is Jeff from Kemin.
Right.
I could give a brief anecdote of our experience doing the wastewater treatment.
Okay, yeah, I think that's fine. Basically, to kind of provide an overview in that regard, right?
Sure. So we had commercialized the technology to use algae, including Euglena, to treat organic wastewater. So those with very high nitrogen, phosphorus, and organic carbon. And while the technology could work, and we have a patent on it, it turned out that really, unless you're doing something very specific, like a targeted heavy metal removal, in general, wastewater treatment systems that are bio-based actually do better with a ecosystem approach because you have different species that are able to do different specialties. And so Euglena could be part of a consortium like that, where you could combine perhaps even different Euglena species, but focusing in on a monoculture approach to treating wastewater. We've tried it and it's more challenging. And in fact, there's other more cost-effective ways to do it, but it could be part of a solution, but a monoculture does not work on its own very well.
Okay.
So when you try to do it with multicultural scenarios, how did you prevent from your environments turning from benefits to harmful? Like
so,
sorry.
Yeah, that's a great question. So it wasn't so much that it became harmful, it's just that no single species is great at doing everything. And so unless you have a very specific and single target molecule that you're trying to remove, let's say heavy metal, in many cases, you're going to have a variety of different objectives. And it might be the case that Euglena could be good at one particular job, but an ecosystem approach is probably going to help you accomplish all the targets. And so for wastewater treatment, you might have multiple different objectives. And so, but that's, what we encountered was that Euglena can do a good job and you can keep it pretty close to a monoculture. But if your job is to treat wastewater, then you have to focus on that goal first. And we found an ecosystem approach worked better.
Okay.
And I'll just add to that really quick. And it may not be that the whole organism, a live organism, is the best approach either. It could be that components from these organisms are more useful than maintaining a live culture. So there's different ways you can go about it, as Jeff said. But yeah.
Okay. Sorry, I don't know if Kengo Suzuki is on the call to contribute to this point. I think Kengo. Okay, I don't think. All right, Wade, you could continue. So I was thinking he's around to also contribute.
I think Kengo just come back.
Kengo Suzuki.
Hi.
All right. Okay. Yeah. Could you please contribute to this as well? I mean, with regards to the question around the pollutants removal and things like that. So far.
So the question was, is there any company using Euglena to remove any pollutants?
Sorry. I'm not sure. Not sure.
I guess not. I guess we don't know that.
Okay. Someone is asking, what about the use of the sensors and rapid monitoring program? So
yeah, I was wondering, can this more specify on it? Give more detail on his question.
Yeah, I think Antonio, you could maybe use the voice system to.
Yes, during the presentation, there was a hint about the fact that Euglena could be used for sensors and for monitoring, and we've not heard.
I think that's right, Antonio. I didn't go deep into that because obviously the biology there is very specific for the signals that are being received, but nevertheless, it's quite a limited machinery, you know, in terms of total number of proteins and secondary messengers that are involved in those transduction pathways. It's very finite. So some of it is better worked out. And you were talking, I think, earlier, weren't you, about how phototropism works for Euglena. And so I think Euglena could even almost unmodified be used as a sensor for a certain stimuli, but in order to translate those into a kind of a microbot, we have to have really good definition of the mechanisms for each of the sensors that are going under. But that's entirely possible within a challenge like this, that research could be funded.
Thank you.
Then I think Sergio has another question. What happens with Euglena biomass after, would that be removal of heavy metal, remediation of heavy metal, contaminants?
Is that Geoff or Scott can help us answer that? You may be able to use it. So from Barry says, you may be able to use, excuse me. Sergio's question was what happened with the Euglena biomass after remove of heavy metal contaminants? Barry's answer was, you may be able to use Euglena to bioaccumulate and then extract the metal from this Euglena or Euglena cell-free extract. In terms of mining, this could involve removing heavy metal, but also reclaiming mined metals.
Metals. Sorry.
Oh, mined metals.
Metals, yeah. Typo there, sorry.
No worries.
So
any more questions? Sorry, Kevin.
No, I was just commenting that this is widely used for various microbes for reclamation of toxic.
Yeah, it's not unique to Euglena, but it is certainly something that could be used.
Barry, do you want to maybe throw more light on that in terms of what the mechanisms and things might be?
Well, yes and no. We are doing some work looking at compounds that bind heavy metals that are produced by euglenids, and using those either in the cells or in cell-free extract. And this is related to what Scott was talking about. So I want to make sure that we don't get a crossed messages here. But one of the things that we would be looking at then is that the metals would be bound by either sets of compounds produced by Euglena, either presented in a cell-free extract or some other form, and then use hopefully some relatively straightforward chemical shift to bring those metals back off of the matrix, which was Euglena based, so that we could recover them. And so the idea would be, yes, we could apply this to removing things like mercury from the environment, but a goal may be to reclaim certain metals or compounds from the environment. So I think that It's just right now it's a matter of identifying the compounds, figuring out how to stimulate the Euglena to produce more of those, and then utilizing that.
All right. Thank you, Barry. I think we're just on time for this grand challenge. I would hand this over to Mark Field and Kevin. Mark and Kevin.
Okay, so apologies, my internet connection went down halfway through the the last talk, so hopefully it will stay stable for the final talk in this session.